So it's my pleasure to introduce Dr. Edwin Aroke, who's going to be talking today for our CAPPI Chat on the role of epigenetics in racial pain disparities. So with that, I'll turn it over to Edwin.
All right. Hey, everybody. So I'm Edwin. I am a nurse and anesthetist by training, and that's why I'm dressed the way I am. And my research really looks at the role of epigenetics in racial disparities in pain. So I'm going to give kind of a big overview of why we're using this approach and what we're finding so far. So this slide really gives us kind of this idea of the pain life cycle and the impact of chronic pain. The idea really being that, from birth through adulthood, we develop all these factors. We are exposed to different risks and resilient factors, which may, be predispose us for pain or may really be protective towards pain, right? And at some point in our lifetime, we all are going to face acute pain. It's inevitable to happen. But for some individuals, unfortunately, they move on to develop chronic pain, right? Despite treatment. Then they may stay in treatment forever or may hopefully recover. So the key is really, why is it that some people, one, go from acute pain to chronic pain, but two, why is it that some people are more responsive to specific treatments more than others? Why do some people develop disability from chronic pain? Those are really some of the key questions that we are trying to really delve into. Specifically, we realize that African Americans and people of low socioeconomic status are disproportionately affected by chronic pain, right? They experience more severe and chronic severe and disabling pain compared to white and non-Hispanic whites specifically. And that is a very interesting thing because one, when you think about race, we all have come to believe now that, race is truly a social construct. So how is it that this social phenomenon, this arbitrary construct that we have developed as humans, becomes a predictor of how somebody feels or the amount of pain that somebody experiences? And I make this assertion or support this view of race as a social construct because to date, we have not been able to really identify any gene or chromosome that codes for race. If you look, for instance, there have been cases of siblings who one looks more lighter skinned than the other, and arbitrarily may decide to identify as white, while another may choose to identify as black, right? And all of these stems from this one drop rule, which just for some historical context, where basically it was that, hey, if any of your ancestry or any of your ancestors had even one drop of black blood, you were considered black. And that real historical context has really stayed with us and affects the life experiences or the lived experiences of individuals. So I truly believe that when it comes to racial disparities in pain, one of the biggest drivers is really the lived experiences. And as such, I'm going to go away from really thinking about, race as a biological factor, but really focus more on the lived experiences. And talking about pain, low back pain is really one of the most disabling chronic pain conditions that you can think about or think of. Again, just like most acute pain conditions, it's true that about 80% of the population are going to experience back pain at some point in their lifetime, right? The good thing is most people will recover, right? But again, if you think back to that pain cycle that I mentioned a minute ago, why is it that some people recover and others go on to develop persistent pain? If the pain lasts more than 12 weeks, we say that person technically now has chronic low back pain. And even within this group, a vast majority of the individuals we really do not know what causes the back pain. It's not related to trauma, it's not related to infection, it's not related to cancer or degenerative disease that we can easily see, such as spinal stenosis or any of those things. So, and within the low back pain population, unfortunately, again, we see the same pattern that we see with most other pain conditions, which is racial minorities and individuals of lower socioeconomic status tend to have more severe and disabling chronic low back pain. So to, again, just as we struggle to tackle this question, we wanted to see within our own population here within the Birmingham area, does this hold true? So in this small study that we did, we recruited non-Hispanic whites and non-Hispanic blacks with chronic low back pain and compared, pain severity and pain interference, right? Because if you want to study a phenomenon, one thing we really wanted to do is ascertain that what we see in the literature still holds true in our sample. So what you see here is truly, yes, there is a significant difference between blacks and whites. Overall, blacks have report or experience more severe chronic low back pain compared to whites and more pain interference, meaning the low back pain that they experience interferes with their activities of daily living, right? With their enjoyment of life, their ability to care for themselves, take care of their loved ones, go to work, things like that. So what is it that is driving this? So one of the first things we did was use psychosocial variables, which is what most of us really do. Look at things like social status. Is it because of somebody's social status that they are experiencing, low back pain more than that blacks are experiencing more low back pain? Or is it because of depression? So we use this moderated mediation model. And basically what we are showing here is subjective social status, which is SSUSM, right, is a strong predictor of pain interference, such that, an individual with, high socioeconomic status or who sees themselves as higher in the social ladder tends to have less pain interference in their life, which if you think about it, logically, it will make sense, right? Those people who think they're higher up may have better access to care, may have, better healthcare overall, maybe more insurance or living in a higher influenced neighborhood. But interestingly, when we included depression in the model, we realized that, well, depression may actually be the mediator, basically saying that that relationship is explained by depressive symptoms quite a bit. But when we now teased in the influence of race, we saw that that relationship actually differed whether somebody was Black or white, such that for Black individuals, having a high subjective social status in the way they see themselves in society led to more depressive symptoms and more pain interference. But for white individuals, as their subjective social status increased, depressive symptoms decreased and pain interference decreased. So we see this change in how these predictors are influencing pain. So when we're looking at what this is telling us is when you're looking at low back pain and pain disparities, it's not just a socioeconomic status thing, right? There must be something going on within the lived experiences of Black people. And the next question we really wanted to delve into further is, what then is it that is going on at the molecular level? What is the mechanism by which all of these things are interfering with the lives of Black people to cause them to experience more pain? So as we know, most of us, right, we are basically a combination of our genes and our environment, right? And as such, we wanted a model of what is it that is going to bring together this? And that is really where the idea of epigenetics comes in. And really, if you think about it, epigenetics literally means beyond genetics. It means it's basically going beyond genetics to see how does the interaction of the environment influence how genes are being expressed, right? And this idea has been around for quite a long time. Initially, it started, by trying to just understand how is it that tissues differentiate, start from the same genome, and differentiate into different tissues. And now this field has really grown exponentially over the last 10 to 20 years to really give us a much better understanding of how environmental exposures influence which genes are turned on and which genes are turned off. And that in a nutshell is really where we are going with this idea of epigenetics, specifically in terms of racial differences in low back pain. And when we talk of epigenetics, there are two main types of epigenetic modifications, right? So DNA methylation is one of the most studied one. Basically, It's the addition of methyl groups to CPG islands on our DNA. Again, keeping it very, a very high level view of it, it's basically modifications on our DNA, such that we do not change the DNA sequence, but basically add these methyl groups to it. Once the methyl groups are added, it affects whether the gene is going to be able to be read and transcribed or not. Another very common type that is frequently studied are histone modifications. Histones are basically these proteins through which DNA wraps itself around. And when you have these histone modifications, basically it impacts or affects how easily that DNA can be read so that we can produce proteins. As a result, again, if you have histone modifications, we are looking at, is this DNA active? Is it being read? Is it being expressed? So those are basically the same words expressing the same thing. So moving forward, just to give you a quick example. So if you look at, for instance, when I mentioned DNA methylation, right? So most of when we're looking at DNA methylation, we really focus quite a bit on the promoter region of genes, right? And within that, we look at these islands called CPG islands, which are basically cytosine guanine islands, a bunch of cytosine and guanine islands with phosphate groups, such that if we have CPG's methylation in the promoter region, generally that will lead to gene suppression. Then we say, well, basically it looks like, for instance, hey, there is something preventing these genes from being expressed. So for instance, if we have the methylation or the presence, it prevents the binding of transcription factors, such as the CAPP2 transcription factor. So if we have methylation present in the promoter region, generally speaking, then we think, okay, this gene is probably silent. So even though you have this gene present, it may not be transcribed, so it may not be doing what your body expected it to do in a simple sense. Histone acetylation, again, too, is deacetylation, on the other hand, too, is equally always almost associated with gene silencing. Now, the reverse is true. However, it's always important to mention that these are general rules. These are not written on rocks, right? If you now go away from the promoter region to other areas of the gene, for instance, the gene body, DNA methylation is not always going to lead to gene silencing. So it's always important to go back and look at specifically gene expressions so that we can actually say, okay, are these DNA methylation changes affecting gene expression before we can really make strong conclusions about this. So why epigenetics, if you might be asking? Again, I think the life cycle of a butterfly really explains this much easily, if you so to speak. So if you think about the butterfly, it goes from the egg to the larva and then becomes a caterpillar and then transforms into a butterfly. But overall, throughout all of these changes, the DNA of it hasn't really changed. It still has the same genome. What has changed over time is basically which of these genes are being expressed and which of them are silent. So that is really what we're talking about, such that if you have two individuals, then, exposure to specific factors affect how we look. So in this case, we have, the first image here was taken when they were 6 years old, right? One of the kids had leukemia, so was exposed to chemotherapy and all of that. You see that even though they were homozygous monozygotic twins at birth and identical twins, so to speak, at birth, because of exposure to the chemotherapy, now their outward appearance has changed dramatically. Mark and Scott here again spend a lot of time in space, right? 1 year in space, and that environmental exposure, that influence of being in space for a year began to change how he looks phenotypically. Right? So we realize that yes, even though at the genetic level, they have the same gene, now their phenotype, their outward look begins to change. The same thing you see here. Again, these were identical twins at birth, but at 50, you can clearly see that one might have lived a much more difficult life compared to the other. And that affects, again, which genes are turned on, which genes are turned off, and that now changes how you may respond. So in terms of pain, we may be thinking of, okay, are there some genes which could have been protective or which may enhance pain, which are turned on or turned off as somebody grows across the lifespan? So most of what we know today in epigenetics really started with these basic studies. And here, in the first study here, you had pups born to an attentive mom, right? And some pups were born to an inattentive mom. And researchers really wanted to examine, okay, does maternal care affect how much stress they experience when they grow up? And sure, they realized that when they develop to adult pups, their genes, and they look at the hippocampus gene, the hippocampus for gene methylation, they realized that pups which were raised by the attentive mom had less methylation and less stress, while those whose mom did not really care had more methylation, more stress. So they went back on the second time and said, okay, what if we switch the pups at birth? Give the pups from the attentive mom to the inattentive mom, and the pups from the inattentive mom to the attentive mom, and let them raise those pups. Again, when they grow up to be adult pups, they realize that, again, being raised by an attentive mom resulted in less methylation and less stress in the hippocampus. And the hippocampus, compared to being raised by an inattentive mom, resulted in high methylation and high stress. So they took it even a step further to say, hey, what if we now took the inattentive mom, administer them a drug that is going to prevent this methylation from taking place, right? Would this affect the pups as they grow? And again, it shows that when methylation is removed, they show less stress, less methylation. So again, just for a full disclosure, I do not make the images, so the idea of the color coding for what is attentive and inattentive is not my making. It's just the images that I found. But I think the key message here is really, our environment, not just what we ingest, but the care and the amount of stress that we may be receiving from our environment may be affecting which genes are turned on and which genes are turned off. So how does that apply to our talk of the day? So if you, for instance, have two individuals, one white and one black at birth, if you look at their DNA methylation at birth, they may be identical. As we go through life, all of us are going to experience some life stressors, right? The stress of education, stress of work, family stress, all of that is given. But now the idea is for African Americans or Blacks, there may be additional stressors, such as the stress of racial discrimination and injustice as they go through life, such that when they now become adults, when you do a DNA methylation analysis or look at their epigenome, you may now start to see differences because of these life experiences, because the lived experiences of Blacks and whites in America may be different from the lived experiences from each other, such that this extra methylation or these differences in methylation may make it so that Blacks will be more likely to develop chronic pain as adults compared to whites. So in this first study, we recruited 50 adults with low back pain and 50 without low back pain. And the idea at this point was, again, establishing some credibility. How do we even start comparing methylation if we do not show that, yes, within patients who have low back pain, there are differences in methylation compared to those who do not have low back pain? So when we did that analysis, we used reduced representation by sulfide sequencing. And basically, this is reporting the different regions where we found some big differences. And these are the top 20 differentially methylated positions that we identified. And if you see the genes which are annotated to those regions, just to give a step back, when we did RRBS, you are looking at about over 30,000, or if you combine them, a lot of differences, right? So it's not just 10 genes that we are dealing with. We are dealing with close to 100,000 genes. So that is why we are reporting here just the top 20. But important genes which have been associated with like cell recognition, cell adhesion, some which have been associated with things like inflammation showed up. So we said, well, we have all of these genes. How do we make sense of it? So we said, okay, let's put all of these genes through machine learning, basically, and identify which pathways are most represented when we look at these genes. And this is what this shows us. When we do functional enrichment pathways, we realize that were pathways which have previously been associated with things like inflammation, right? The fractalkine production, the bumpering receptor signaling. Other pathways equally involved in things like bone formation and chondrocyte development appear to show up, suggesting that when you have individuals with non-specific low back pain, a combination of changes in immune response, inflammation, and equally bone formation may be playing a role in non-specific low back pain, meaning that differential methylation or differences in these pathways may play a critical role. So the question then was, how do we go from this to really saying, well, race is the issue? So we asked ourselves, are there any differences when we looked at the interaction of race and pain within the context of these genes? Does this interaction hold true? And again, we identified several genes which show that yes, there was an interaction between race and pain using epigenetic differences or differences in epigenetics as our outcome. Some of the key genes showing up here again seem to be very of interest to us, such as the SYN3, which plays a role in synaptogenesis. We equally have some of the top two ones here are interesting, but we're still trying to figure out clearly what their main role is in pain. For instance, the PAH gene is for phenylalanine hydroxylase, which again, it's a very important enzyme in the body, plays a role in the conversion of this amino acid, but we're not quite sure yet where it fits in with pain. And just to give you kind of an example of when we say an interaction, basically you see here with the phenylalanine PAH gene here, in blacks it's showing that, hey, for when you look at blacks, healthy controls, you have less methylation compared to blacks with low back pain. But in white, the healthy controls have more methylation compared to whites with low back pain. So telling you that the methylation patterns in blacks and whites for this gene within our sample is not the same. And that is the same thing with this gene. So we then went back and really said, okay, let's try to tease this even further and really dig into what if we were to just look at individuals who have low back pain, right? And say, let's say within patients who have chronic low back pain, how does the methylation difference look like? And which pathways are being differentially methylated? And that is where we compare 25 blacks and 25 whites with chronic low back pain. Again, these are the top 20 positions which are differentially methylated. You see here a lot of regulatory genes which code for a lot of regulatory functions, such as the long non-coding RNAs, the zinc finger proteins, all of these. But equally, you have other genes such as the neurotensin receptors and equally membrane genes showing up. So when we put them again through machine learning, basically looking at functional pathways, we realize that when you look at, as it applies to low back pain, when you compare blacks with low back pain and whites with low back pain, some of the key pathways which are enriched by differentially methylated genes include those involved in axon guidance mediated synaptorin, wing signaling, cadherin, G protein signaling, the hedgehog histamine, and all of it. So what does this really mean, basically, in a nutshell? Well, we realize that wing signaling, cadherin, and axon guidance, these are all cell recognition pathways. But more importantly, they have been historically they have been shown to play a role in neuronal development. The G proteins, as we know, play a big role when you're thinking of transmission of pain signal, pain transmission, and equally the action of some of the catecholamines. Histamine 2 relates to most of the inflammatory response and the response of our body to inflammation. So what we are beginning to see says, hey, the story that we saw earlier with patients with low back pain versus those without low back pain continues to hold true, which is neuroinflammation may be the reason why we are seeing racial differences in low back pain. So do all blacks and all whites look the same, right? Why is it that then some blacks have pain while other blacks do not have pain, right? And that was the next question we wanted to really tackle. So basically going back to say, what if we were to separate blacks and really take a big closer look at blacks with low back pain versus blacks without low back pain? Do we see any difference? And this gives us a top 20 differentially methylated loci here. And again, you have genes which are of interest, such as the myosin genes, the pexin gene, again, synaptogenesis, neural navigation, genes associated which play a big role in regulation of transcription begin to again show up. So because we had so many of these genes, to make sense of it, we say which pathways are involved, what pathway is being differentially methylated, and which pathways are not. Okay, now I see, yeah. So this table gives us the same thing when we ask the same question for whites, basically saying, when you look at whites with low back pain versus whites without low back pain, which areas are differentially methylated? Again, we see things like cadherin, which play an important role in cell recognition and adhesion, complement factors, HISTRA-A, again, genes which previously have to an extent been associated with things like inflammation, cell recognition. Basically, how do the cells communicate with each other and recognize each other? Which pathway? So these two images here give us a comparison of the different pathways when you look at pain in differentially methylated genes in blacks versus white. So the top one shows you enriched pathways in blacks from differentially methylated genes, compared in blacks with pain with blacks without pain. While the bottom one is showing you the top enriched pathways in whites with pain compared to whites without pain. Notice that one thing we found really interesting was the amount of significant pathways in blacks. So again, the axon guidance mediated by the semaphorins, the cadherin, the G protein signaling, vitamin D metabolism, all of these were statistically significant in white, but most of them are not statistically significant in white. So what the story appears to be telling us now is, when it comes to racial differences in chronic low back pain, multiple pathways appear to be differentially methylated in blacks who have low back pain compared to blacks without low back pain than they are in whites. Is this causality? No. But I think it gives us a clear picture as to what the next steps could be, which is, can we now start looking at some of the genes within these pathways and really identify whether these genes are differentially methylated? The other thing we've equally tried to do is really take a closer look at, okay, within these pathways, how is this related to pain? And this is just an example. So we took, for instance, the beta-catenin, which is involved in most of these pathways, like the Wnt signaling pathway. And you realize that this has been described quite a bit in something like depression, where you see that in a normal individual, right, beta-catenin helps in stressful situations, so that when they experience stress, beta-catenin is not phosphorylated. So without phosphorylation, it can accumulate in the body, leading to stress resilience, expression of more protective genes in the body, and those individuals can better handle stress. But for patients with chronic depression or who have experienced chronic stress, beta-catenin does not accumulate because it binds to this complex, right? Leads to less expression of the genes which are supposed to be more protective of stress. As a result, which my screen here can show me. As a result, you see that when you compare normal individuals with depression, you see that stress-related behaviors such as depression and memory impairment become much more evident. So basically what the story is saying is, yes, as you start paying closer attention to what these pathways are doing, stress may be playing a big role. Maybe what the common thread is really the fact that the amount of stress that patients with depression or when they're under enormous stress and chronic stress for a longer period of time, these pathways, the differential methylation of these pathways may be what is really driving how much of these behaviors that we see. Can I say categorically that that is what is happening in pain? No, but we know very well that something like depression has a very strong association with chronic pain. Most patients with chronic depression tend to have some level of pain, while most patients with chronic pain have some level of depression. So I think we can make some association there, but again, something that we'll definitely explore down the road. So what are some of the next steps that we are taking? So the next steps are really, we want to now take this and replicate it in a bigger sample. We want to be able to do this work now in a bigger sample, but more importantly, we want to validate this, which basically means we want to collect more samples from individuals, take it to the lab and really pick out some of these critical pathways, look at genes within these critical pathways and say, when we have a differentially methylated loci or differentially methylated gene, does that differential methylation actually affect gene expression? As such, we'll be looking at not just epigenetics, but equally transcriptomics, so that we can truly say, yes, when we see that there's differential methylation within this gene, it is truly affecting gene expression, and even take it a step further to say it's affecting which proteins are produced, so that we can now have better targets for pain medications in the future, right? So basically, in a nutshell, what the stories seem to be saying is, yes, we know for now, African Americans have different life experiences in the United States, which may be more stressful than that of whites. And maybe, these experiences may be what is causing them to experience more severe and disabling chronic pain. In a simple sense, epigenetics is just one mechanism by which these experiences, which we call environmental exposure, get under our skin. So maybe the chronic stress, the chronic exposure to discrimination or injustice, or the difficult lifestyle that they live, make it so that some genes are being expressed while others are being silent. These genes may be the ones which may be promoting more pain, or which were supposed to be protective but are no longer protective. Alterations in genes expressed in pathways involved in neuronal formation, right? And again, some involved in inflammation appear to be playing a role when we think about racial differences in pain. But like I've mentioned multiple times, this is an ongoing area of research that we'll continue to explore, and we'll be exploring even further. So of course, this work will not be done without a very supportive team. Dr. Borrell is my mentor in Disparities pain and has been very helpful. And our work has been supported generously by the ANA Foundation. The NIH supports our work, the School of Nursing, and Minority Health here at UAB. All right. Thank you.
That's terrific. So let me ask you, now, of course, we can take questions from anybody, but I'm going to just jump in right here because I unmuted myself first. So, I mean, it's really, really cool to see all these different genes and to, try to sort out, what they might be doing and why they're up or down in different groups. Now, you were looking at, comparing blacks and whites with pain, and then within each group, pain versus control.
Yes.
So have you looked at differences between blacks and whites in the healthy controls? Because any of those differences might be, sort of getting at the psychosocial stress aspect. I mean, because these individuals don't have the pain side, but all the other environmental stressors will still be there.
We haven't looked at it, but I think definitely that would be an interesting analysis to go back and look at, to say, okay, and it may really actually help us explain some of the differences that we see, because one question we continue to have is, how do you know that this is what is causing pain versus just the fact that somebody is black or just the fact that somebody is white, right? So I think for sure that would be a good analysis to look at, to say, okay, in our healthy controls, what differences do we see? And definitely doing that and now comparing that with patients with pain would really help, maybe begin to tease out even the pain differences much more.
Mm-hmm. Yeah. So, I mean, I was of course really interested that, in some of those tables, when you looked at where those genes are clustering, one of them was Th1 cells.
Yep.
Now, obviously, I have a vested interest in immune cells and pain. But do you remember? I mean, were those genes, would they be more active or less active in pain? And usually, methylation would be turning them down, right?
Okay. Methylation will be turning them down. So that is I have to look at that gene specifically, because the interesting thing when you do RRBS is you are saying, well, in this person there is more methylation compared to this other group, right? Now, to be much more clear, if we want to really answer whether it's hypermethylated and regulating it, then we have to do what Dr. DeBerry does, which is do the wet lab analysis of it. That is really when we can clearly say, okay, as it relates to this gene, it has some regulatory, or the epigenetic change that we are seeing truly has regulatory functions, which is why we tend to go with pathways. Because when you have changes, you say, okay, this cluster of genes, where are they all coming from? What pathways are they most involved in? Because what we have done is basically say, look at this globally and just tell me what regions or which genes are playing. What are the key players here? To be able to answer that quick question, to say, okay, is it truly methylated? And then I will have to say, okay, what region is it, right? So is it in the promoter region, or is it in the intergenic region of the gene? But that is something I can easily answer going back to that table, I think. So you said which one was of interest? And that would be just a preliminary view of it, because a true answer on it requires the wet lab analysis, which is our next step.
Yeah. So I think one of the really cool things here about this sort of epigenetics is that, when we look at sort of behavioral outputs, or, just, cytokines in serum or something like that, when we're looking at inflammation, when we see differences, we just assume that, well, it's a different process that's happening. There's more inflammation here than there. But if you're also, if one group that shows, say, less inflammation also has a lot more methylation, I mean, it may be that the processes are the same, but there's more of a break on one system.
On one system, yes.
And so, then it sort of changes that interpretation, is that both groups have got the same machinery, but there's been alterations to it. It's not that they're two different sets of machinery.
Yes. And so one analysis that we are currently trying to finish up is really trying to answer that, where we are looking at some of them in cytokines, the inflammatory cytokines, and now stepping back to this sample to say, okay, how are the genes which code for these cytokines behaving? Is there the same direction in terms of, okay, if you look at, for instance, IL-6 or IL-2 and say, well, we would expect that if you have an increase, we should be seeing an epigenetic change, right? Like we say hypermethylation, which is saying, hey, this gene is being expressed, right? We should see the same story, such that the increase in inflammation that you see should be something that we should be able to detect epigenetically. Granted, because technically that is really what the body, that when you think of physiology and pathophysiology, that is really what the body does naturally. Each time you need a protein to be produced, right, there is the instruction, it's delivered. It says, hey, we need this gene, we need this protein. So the genes coding for those proteins are turned on or turned off, depending on how much of the protein is needed. And that mechanism of turning those genes so that the inflammatory cytokines are produced is epigenetic.
Wow. So if a gene is expressed more often, will it have more methylation? Or is it more likely?
No.
Okay.
It's expressed more, then you expect the promoter region to be less methylated. Methylation will lead to silencing if you have more methylation in the promoter region generally. Then you should see less methylation in the promoter region of those genes. Because methylation interferes with the binding of transcription factors. And when the transcription factors cannot bind, that gene cannot be transcribed or read, so it will be less expressed.
Okay. So I was curious because, I mean, when we're sort of at a bigger level, I mean, if there are a lot of one neurotransmitter constantly being released, then the receptors can be downregulated as a result. And
yes.
So, if IL-6 is constantly being released, does the body, adjust by methylating some of those genes just to turn it down in that sense, rather than at the receptor level?
So basically what you are saying is, does upregulation or downregulation, is that an epigenetic modification? I would argue, yes, it's an epigenetic change. But now in this case, you are responding, it's similar to, because remember, things such as stressors, right? Glucocorticoids generally, and stress hormones, for instance, but their basic mechanism of action is such that they work at the receptor by regulating, the nuclear material. So basically, for it to work, it has to go to the nucleus and tell the nucleus, hey, produce this thing, right? And tell the enzymes that, or the genes that want this produced. So when we say there is upregulation or more production, that is really what it's saying. It's saying the machinery is saying, keep producing it. Similarly, if you have somebody who has muscles which are not being used, for instance, right? And you're telling the body is saying, hey, we don't need this, or we need more of it. There has to be that epigenetic modification taking place. And that is basically how the whole concept of epigenetics works. And going back to what I mentioned earlier, which is, if you think about tissue differentiation, right, where when you start out from the zygote during fertilization, how do we go from that to now having specialized tissue such that the neurons are so different, for instance, from skin cells? It is that differential methylation, it's that differential epigenetic modification, right? So that is really what it is. So that basically, even though we don't talk so much about epigenetic change, and methylation, it's important to clarify, is just one type of epigenetic modification, right? So we have so many other types of epigenetic modification. I didn't mention histone modifications. You equally have like long, this microRNAs and all of this. So there are other types of epigenetic modifications that take place in the body. So it's not all just DNA methylation. The only reason we've studied DNA methylation the most is just that, one, it's relatively easier and more stable. Plus now we've seen that, yes, they are heritable. As a result, it has a lot of interest. Plus, equally, the fact that they are reversible makes it even more appealing to study, right? Such that if we know that, okay, these factors are increasing methylation, we can try to reverse some of that methylation.
Very cool.
So I have a quick question, Edwin. I don't know what the age of your study participants was, but is there a possibility that you could study any of their offspring who may or may not also have chronic pain?
So the age of the participants, all the participants are between 19 and 85. So would we be able to include offsprings? Maybe. Larissa, what do you think? Can we recruit offsprings?
I think you could. It would just be a matter of gathering them.
Yeah.
That would be the challenge.
So maybe we can see if some of these are heritable.
Yeah, it would be really interesting. Or even just a survey of how many of the offspring do or don't have chronic pain from each group.
So Salva says, were the subjects from different socioeconomic strata? Yes. So there were, the subjects are from different socioeconomic backgrounds. And when we look at socioeconomic status, does not really explain the differences within this sample when we look at socioeconomic factors. And just to go to the the heritable part of the epigenetics, other studies not in pain, I haven't seen any transgenerational studies in pain yet, but when you look at other fields like depression, PTSD, there have been studies looking at transgenerational epigenetic changes. And they have been shown that yes, when moms experience so much stress or depression, that affects the methylation patterns of the offsprings. And some studies have even looked at the second generation. So truly, some of these changes are believed to be heritable, even in humans.
That's awesome. And I mean, I think, sort of to to piggyback on that idea, I mean, those studies, that are looking at stress and stuff like that have those transgenerational effects. I mean, we know that stress interferes with with pain and contributes to pain. And so if those are transgenerational, then yeah, I mean, it's quite possible that it is going to affect the offspring.
Yeah. So another question I'm seeing here, did you look at pain relievers? We did record the amount of pain or whichever pain medications they take and not take. The only thing is it's so sporadic. And when you look at the cell count, it's usually very small. So we would not control for that, which is something as a sample size increases, we will look at it because there is the emerging field of pharmacoepigenetics. The idea too that as we take medication, so for instance, if you remember back to the kid with leukemia, that was an example of a pharmacoepigenetic, right? Where the chemo treatment affected her phenotype, how she looks. That's why people lose their hair and things like that. So, and there are equally studies already looking at opioid exposure and saying, hey, does exposure to opioids for a long time lead to epigenetic changes, which may be one leading to more addiction or less? I mean, that's a whole area of its own, but that's one active area to the pharmacoepigenetic aspect of pain. So controlling for pain will be good.
Yeah, and you could do that with some of the diet data that Burrell has too. That's all done because there's nutrigenomics.
Yeah.
Yeah, the effect of like food turning on and off. So feel free to use that data because I want people it took me a while to get it. So I'm like, yeah, use it, please.
Yep. So it will be good to look at some of those differences. I think the key in the future is really going to be being able to look at multiple omics, right? The multi-omics. Because, for instance, in this data, we do not really look at the genes themselves. So we are not even talking about whether you have this gene or not. We are just saying, is this gene being turned on or turned off in your body? How is the methylation looking? So if you truly want to be able to understand the mechanism, first we have to start by saying, what genes do you have? So that we can look at things like polymorphisms within those genes, because those equally affect whether the gene is going to be able to be expressed or not, and control for that. Then look at the epigenetics. Then maybe at, like Larissa is saying, what does your nutrient look like? What drugs are you taking? All of these layers are different omics approaches, which if you want to do that, then the sample size will be in the thousands before you can even start, touching a little bit on it.
So has anybody looked at like, certain SNPs and then versus like epigenetics?
The good thing with epigenetics now is we know for the genes, we know some SNPs which are notoriously known to affect whether transcription will take place or not. So when you are doing the analysis, you control for those kind of SNPs to say, okay, let's just get rid of this and make sure that we are not trying to look at a region that a SNP may have been the problem. Yeah.
So I'm thinking, in some of those big like genome-wide studies, they show that, people with certain versions of a gene, have less pain, or, are more susceptible to pain. Yeah. And so in that case, having, a SNP that makes you more likely to have pain, having that be methylated maybe isn't a bad thing.
I don't know, maybe that would be the next step. Dr. Diber, what do you think? Does having the SNP affect whether epigenetic change is going to take place?
Yeah, sounds complex. It's really interesting to think about, though.
Yeah.
Yeah, that's real precision medicine.
Yeah. But I think for sure that is the wave of the future. One of my personal interest in looking at more epigenetics than the SNP was that aspect of what then can we do, right? If I see that you have this SNP, what can you do about it? Versus, with epigenetics, at least there is some hope. If we see that, hey, chronic stress is causing all of these things, maybe we should be more aggressive than giving you medication. Let's be more aggressive in reducing your stress, and maybe some of these things that we are seeing would, reverse themselves. So the dynamic nature of epigenetics to me is very appealing compared to, the static nature of genetics, where we just identify it. So what? Yes, we can prevent some, we can, say avoid some medications like in pharmacogenetics and things like that. But overall, the appeal of epigenetics is really that dynamic nature. The fact that what you eat, affects it. The fact that how much stress, how much you exercise and all of those things affect your epigenome is, to me, very important and relevant to the conversation of race differences, right? Because if we are seeing that chronic stress is a problem, then maybe as we think about racial differences and trying to promote health equity, we can be more active in saying, hey, we really have to stamp some of these systemic racism and all of these injustices that we see.
So there's one more question in the chat, and then I think we'll have to call it.
Did you look at the presence of other chronic diseases, obesity as a factor? Dr. Sauce, we are looking at obesity now as we speak. Yeah, so we look at other chronic diseases, but the key thing that we are looking at now is obesity. That's an ongoing study in the lab. So stay tuned for that.
Yes. Sometime soon.
Yeah.
All right. Well, thank you very much, Edwin, and everyone else. Thank you for showing up and for such a great talk.
Thank you, everybody.